In this video, we're going to learn two ways to solve dihybrid crosses. The first way is going to use something we already know, which is the Punnett square. In a monohybrid cross, the genes that the parents are carrying separate away from each other, and we put them on each of our rows and columns to represent how the genes are distributed to the offspring. When we did this, we figured out the gametes without actually knowing that that's what we were doing. So in a heterozygous pair, there is one gamete that's a big A and one gamete that is a little a, meaning two types of gametes get made by that parent. If it's a homozygous combination, however, like this homozygous dominant, there's only one type of gamete that gets made because all of them are big A. When we start working with a dihybrid, we have two genes to consider. So where in this case, the big A and the little a would be found in different gametes, the same thing would be true of the B. So we end up with a combination that would look like this. So one gamete's going to have a big A, big B, and the other's going to have a big A, little b. With the heterozygous parent, the big A is going to go with the big B. The big A will also have a possible combination with the little b. The little a will go with the big B, and the little a may also go with the little b. In each gamete, there has to be one A gene and one B gene, and when you work out all of those combinations, you end up with 4 possible combinations. You can see with our larger genotypes where we have a trihybrid or even more genes than that it gets a little cumbersome to figure out how many gametes you need to have. So of course we're going to have a rule to help us out with this. It's called the gamete rule, and it's 2 to the n, with n being the number of heterozygous pairs. So in the case of this genotype right here, we have 2 heterozygous pairs. That is 2 to the second power. That is 4, which means we can expect 4 gametes from that. In the bottom example, we have 1, 2, 3, 4. That is going to be 2 to the 4th power, which is going to be 16. We can expect 16 gametes. In a dihybrid trait, we only have 2 genes, so we don't really need to worry about any more than 4 gametes. So if we look here, it's going to start out like any problem. We're going to need to have a key. The key's already done for us, as it's already done in your notes. And the second thing that we need is to know what our parents are, which is shown right here. So we're going to be crossing big A, little b, times little a, big B, little b. First, I need to know what the gametes are for my 2 parents. So if I look at this parent has 1 heterozygous pair. That is 2 to the 1st power, which means that I can expect to get 2 gametes. Now, as I do this, I'm going to take a look at each of these and make sure my gametes are mapped out. So this parent right here is big A, little b, right? The second one would also be a big A, little b, so I don't need to write that. It's the same pair. The next one is little a, little b, and again, I don't need to write the second one. So I have 2 gametes as the 2n rule told me I would. Same thing here. This has 1 heterozygous pair. It means that I can expect 2 gametes. So in this case, it's going to be a, big B, and a, little b. Now, unlike a monohybrid cross where we always put 4 squares down every single time, in the case of a dihybrid cross, we really only need to build the square that we need to complete. So in this case, one parent is going to need 1, 2 rows. So I'm going to put big A, little b, and little a, little b, and that's my 2 rows. My other parent is also going to need 2 spaces, so that's going to be a little a, big B, and a, little b. And now my square is set up. Now the next part of this is simply lining this up the way that I would with any other Punnett square. So this is big A, little b, big B, little b, little a, big B, little b, big A, little b, little b, and little a, little a, little b, little b. My genotypic ratio is going to, again, be the same. I'm going to find how many I have that have exactly the same genotype. And if I look at these, if I'm only building the square I need, I shouldn't have too many extra. So if I look here, I've got one of these. So I have 1 out of 4, big A, big B, little b. I have this guy here, which is 1 out of 4, big A, little b, little b. I have this guy right here, that's going to be 1/4, little a, big B, little b. And this guy right here, 1 out of 4, little a, little b, little b. My phenotypes are a little bit trickier because I have to consider both traits at the same time. And I find it easier if what I do is take a look at my key and pair them up before I start counting. So I can get dimples and I can get freckles together. I can get dimples and no freckles. I can get no dimples and freckles, or I can get no dimples, no freckles. Now I have to look at each one and see what I have. In the case of the first one, I've got dimples and freckles. I need dominant, and that's going to be found right here. That's 1/4. I don't have any other genotypes that have dominant in both the A gene and the B gene. I've got dimples and no freckles. That's a dominant and a homozygous recessive, which I have right there. So that's 1/4. I have no dimples and freckles. That's a recessive. That's either one of these. And freckles, which is dominant, that's this guy right here, so that's going to be 1/4. And then no dimples, no freckles is going to be my homozygous recessive, and I have 1/4 there. That takes care of the problem. I've got all 5 parts of the problem demonstrated. Doing the full Punnett square is important if I need to know all of the offspring, both their genotypes and their phenotypes. But there's a second way to solve these problems as well, and that is using mathematical probability. We're going to use the exact same problem to do this, so you'll notice that the key and the parents' genotypes are exactly the same. We're going to set up the A trait separately than the B trait. So we're going to start with this part of it right here, and we're going to set up a monohybrid cross for the parents. And it's just the A's that we're going to do, so it's big A, little a, little a, little a. And we're going to fill out our square the same way that we would if we were doing a monohybrid cross. So now we're going to do the same thing for our B trait. So here is our monohybrid Punnett square and our little b, big B, little b. So again, we're filling this out the same way we would if it was a monohybrid cross. Now, this is where we're going to be doing something different. We're going to be setting up an equation that's actually going to be the description of each of the squares. So we're going to start with the A square, and we're going to describe what we see in the square. So if I look right here, I have 2 out of 4 that are big A, little a. That's half my square. And I also have these guys right here. That's 2 out of 4, little a, little a. That describes my A trait square. Now, because I'm going to be mixing these, this is going to be a multiplication, and then I'm going to also describe my B square, which is these two right here. So it's 2 out of 4, big B, little b, plus 2 out of 4, little b, little b. There's my B square. So I'm going to be multiplying these, and to do this I'm going to use something that's similar to the FOIL method. And to do that, I'm going to multiply these guys together. Now my A's and my B's act like variables, so I would combine my variables, and I'm going to multiply my my fractions. So 2 times 2 is 4, 4 times 4 is 16. So it's 4/16, and I'm combining my variables, big A, big B, little b. So that'd be 4 out of 16, big A, big B, little b. I'm going to multiply these. That's going to be 4 out of 16, big A, little b, little b. I'm going to multiply these. Again, that is 4 out of 16. Oops, there we go. 4 out of 16, little a, big B, little b. And this times this, that's going to be 4 out of 16, little a, little b, little b. Those are my genotypes. My phenotypes, on the other hand, are going to be, again, my combination, and I'm going to do some abbreviating here so I can fit it in. But we're going to have dimples and freckles. We're going to have dimples and no freckles. We're going to have no dimples and freckles, and no dimples, no freckles. And when I combine these out, my dimples, freckles, that's this guy right here, that's going to be, and I'm going to reduce my fraction, 1/4 big dimples and freckles. This guy right here, 1/4 dimples, no freckles. This guy right here, 1/4 dimples, freckles. And this guy right here, 1/4 no dimples, no freckles. And if you compare that to what we did in method 1, our phenotype is exactly the same. Our genotype is the numbers that we got from the multiplication are not reduced like they were when we did method 1, but you can obviously see how that would work. The beauty of the math method is that you can actually use this to do shortcuts so that you don't need to do the entire square. So for instance, in the problem that you see here, if we were mating these two sets of people, we can actually figure out one specific genotype without having to do the entire square. Now, you might be able to do this in your head, but I'm going to do it. I'm going to write out the whole thing to make sure that you understand why we're doing it the way that we are. So I'm going to start off, first of all, by looking at the A trait. If I'm crossing big A times big A, that would be a Punnett square that looks like this. And I am actually only looking for one part of the square. I am looking for this one right here, which is the big A, little a, and I have this right here. So I know that for this particular gene, there is a 2/4 or 1/2 chance that I'm going to get that big A, little a. Now I'm going to do the same thing with the B's. In the case of the B, it is big B, little b, times big B, little b, right? This parent and this parent. So that is going to be a square that looks like this. And what am I looking for? I am looking for a little b, little b. I only have 1/4 of that, so I'm going to put that guy down. I'm going to go to the next one, which is my C trait. That's little c times big C, little c. And when I work that out on a square, that's big C, little c, big C, little c, little c, little c, little c, little c. And here is what I'm looking for. So that is going to be 1/2, of the square, or 2/4 of the square. And then my last one is the big D, little d times big D, little d. So big D, little d times big D, little d looks like this. And we are looking specifically for the big D, which is right there. So that's going to be 1/4. So when I multiply all this together, this actually would be easier right here if I had reduced that, and I can do that because I'm multiplying fractions. So this is going to be 1/64 because it's 2 times 4 times 2 times 4, which is 64. So the chance of having a child with big A, little little b, little c, big D is 1 out of 64. So in order to solve this problem, I would have needed to do 64 squares. This is obviously much faster. In this problem, same deal. This is big A times big A, little a. I'm looking for big A. If I think about the square and the way it's put together, a heterozygous cross is going to be 1/4 big A. If I use my little b times little b, what's the chance that I'm going to get little b? The entire square is going to be little b. That's a 1 over 1. That's the entire square. And you could just write 1. If I'm crossing my big C times my big C, what's the chance I get big C, little c? That's going to be half of the square. And if I'm doing big D times little d, what's the chance I get little d? That's also going to be half the square. So when I do this, it's going to be 4 times 1 times 2 times 2. It means that my answer is going to be 1/16. So hopefully this is helpful, and if you have any questions, just make sure that you ask Miss Chambers or myself, and we will both be very happy to help you.